Hello, welcome to section 2 of module 4, working with EEG data. And this lesson is going to be about EEG artifacts and pre-processing part 1. Note that section 2 is going to be long and it covers a lot of content for working with EEG data, so I hope you bear with me since these technical concepts are going to be very important when it comes to working with EEG data. So let's jump right into it. And here's what we already know. EEG is a form of electrophysiological neuroimaging, and it records the brain's electrical activity. Some key questions are: what are the stages of EEG processing? What is the signal-to-noise trade-off? And what is interpolation, epoching, filtering, and downsampling? Just a side note, so a lot of the topics I'm going to talk about in these sections primarily come from this book by Mike Cohen, which I highly recommend if you want to learn more about analyzing electrical brain signals. It thoroughly explains the conceptual, mathematical, and implementation aspects of EEG analysis, and so it can be very useful if you want to work with EEG. Alright, so these are the steps, the stages for EEG processing. This is not necessarily the exact ordered procedures that you have to adopt, so it's mainly up to you and what your goal is. You can deviate from the exact sequence of processing. The order and which ones you do depends on the specific experiment and the types of equipment you have and so on. Sometimes each stage would be implemented in a MATLAB or Python script, and the first stage would be the steps between the raw data and running the next stage of pre-processing, like independent component analysis, for example. The pre-processing here would be stage 1 and 2, and the second stage would be the final step of pre-processing before you start with the analysis. And in this table, stage 3 would be single subject analysis, and stage 4 is group level analysis. Note that I'm not going to go over all of these methods, therefore what I'll be going over are the ones I think that are most important, but if you want more information about EEG processing, feel free to check out Mike Cohen's book. Alright, and in this lesson, as well as the next lesson, we will be focusing on pre-processing. So here are some of the EEG artifacts and what they look like in an EEG signal. There's breathing, tongue movements, electrical noise, sweat, eye movements, head movements, cable movements, heart rate, muscle movements, and so on. The whole point of this section is to see how we can deal with these artifacts and whatever kind of noise that exists within our EEG data. So, what is pre-processing? Pre-processing is all the steps that you do to your data between acquiring the raw data directly from the experiment and the stage where you're ready to start doing the analysis, like time frequency analysis. So some pre-processing steps organize the data to facilitate analysis without changing any of the data, such as extracting epochs from continuous data. Other pre-processing steps involve removing bad or artifact-ridden data without changing clean data. For example, removing bad electrodes or rejecting epochs with artifacts. And then some pre-processing techniques involve modifying otherwise clean data through the application of temporal filters or spatial transformations. Remember that many pre-processing choices and steps depend on the details of the experimental design, the equipment used to collect data, the analysis you plan on performing, and idiosyncratic protocols and preferences. So it's a good idea to keep track of all the details of pre-processing for each subject, such as which trials were rejected, which electrodes were interpolated, and which independent components were removed from the data. Since some pre-processing choices may introduce biases to the data, you should use the same pre-processing procedures for all conditions to minimize the possibility that any biases would produce condition differences. And as you already know, EEG data contains signal and noise. So appropriate pre-processing will attenuate the noise in the data However, signal and noise are often mixed together and may be difficult to disentangle completely. Thus, there is a trade-off between signal and noise. Removing a lot of the noise would likely remove some signal, and leaving as much signal in the data as possible would likely keep the noise. So your criterion for balancing signal and noise may influence the steps of your pre-processing. So where you set this criterion also depends on how much data you have and on how difficult the data are to acquire. So for example, if you have hundreds of trials, you have the luxury to remove many trials that contain noise. On the other hand, if the data were limited and difficult to obtain, you might be willing to tolerate some noise in order to retain as much signal as possible. And fortunately, time frequency-based analysis tend to increase the signal-to-noise characteristics of the data, particularly for single trial analysis and for relatively low frequencies. Oftentimes, EEG data, especially high-density EEG data, can contain bad channels that provide inaccurate information. So here are a few reasons why a channel might be excluded. You can detect bad channels even before you finish collecting data. For instance, if you know that one or more of the channels were not functioning properly, or if you notice that any of the electrodes lost contact with the scalp during the experiment, you can mark them down and exclude them from analysis. So it takes experience to determine how much noise is appropriate, and using techniques such as independence components analysis, or ICA, can remove some of the noise without having to resort to removing an entire channel. Again, if you have a lot of bad channels, or if you don't have many channels to begin with, simply removing bad channels will result in a significant loss of information. So in that case, you might want to repair or interpolate the excluded channels instead. So interpolation is a way of filling in the missing data based on the other data that's available. After flagging bad channels, it is common practice to interpolate data for the bad channels on the data from the good channels. So the most common interpolation is by spherical splines. The first step is to project the channel locations onto a unit sphere representing the head. Secondly, compute the matrix that describes the relationship between the good and bad electrodes. And third, use that resulting matrix to interpolate the data for bad electrodes. Right. Another step of pre-processing is epoching. So EEG epoching is a procedure in which specific time windows are extracted from the continuous EEG signal. So these time windows are called epochs, which are usually time-locked with respect to an event such as a visual stimuli. So if your EEG data are in a matrix where the rows represent channels and the columns represent time, so each row is a channel, after the epoching, you include the epochs, which is the number of segments you extracted from the continuous EEG signal. If you want to extract epochs from your signal, you should know what are the segments of interest to be analyzed, such as a specific stimulus. Alright, so this is another very important pre-processing step. So we would want to filter EEG data in order to make the data look cleaner and easier to interpret. And it can also be used to reduce some noise. Here are some of the types of filtering that can be used for any types of signals. You might have encountered some of these already. And so in low-pass filtering, the low frequencies below a certain value are kept, while high frequencies are removed. This is also known as a high-cut filter. And next is high-pass filtering, which is also known as low-cut filtering. It only keeps high frequencies, and only those below a certain value are removed. And in bandpass filtering, we basically combine low pass and high pass, and we keep only frequencies between a lower and upper bound. The opposite of this is the band cut filter, which removes frequencies in a particular range. So a popular type of band cut filter that is necessary for EEG is the notch filter, which removes a single frequency. It is possible to combine multiple notch filters to remove a particular set of single frequencies, which is useful for things like removing electricity noise. Removing electricity noise is important because generally the electrical circuits surrounding your measurement will introduce noise at around 60 hertz. Right. So, say that we have an EEG system with 64 channels with a sample rate of 1000 hertz per second or 1000 hertz, sorry, 1000 cycles per second. Anyway, if we represent each sample as a 32-bit float, that's around 2 million bits. So a lot of this information would be redundant, and having this much information could make data processing pretty slow. Therefore, we do downsampling. Downsampling is a technique for reducing the sampling rate or reducing the number of samples used while still trying to maintain the information that is needed. And strict downsampling is when you keep every nth sample. And there's another type called decimating, which is just performing a low-pass filter first before doing the downsampling to remove high frequencies that cause artifacts. Okay. So here is the summary. EEG processing can be divided into a series of stages, and pre-processing is one of them, which involves extracting epochs from continuous data, removing bad channels, downsampling, and filtering out artifacts. Overall, pre-processing cleans up the data and allows us to get a more clear picture of the desired signal. And what's next is EEG artifacts and pre-processing part 2. So I'll see you in the next lesson.